Hello students. Today we are moving further in the topic principle of inheritance. Today as we have studied previously the law of inheritance, we have studied the variation, we have studied clone, we have studied all three laws of dominance, independent assortment and purity of gametes. Now we will going to study the inheritance on the basis of chromosomes. So which is also considered as a chromosomal theory. Okay, so as we all know though, chromosome carries genetic information as it has DNA itself. So in front of you there is a figure by which you will understand the two processes. First is mitosis and meiosis that you have studied in the previous class. In mitosis and meiosis we know that chromosomes used to get divided and form separate cells. In mitosis you can easily see the genetic material is first doubled, then it gets segregated. That is chromosome, and each cell has appropriate amount of genetic material. In the same way with the, if we talk about meiosis, in meiosis the division is there, but in this the cell formed are having used to have half genetic material. So half genetic material which afterwards get doubled. So basically is in chromosomal theory given by Sutton, he clarified that likewise gene chromosome also carries from F1 generation and goes to F2 generation, and it carries the same information which a gene used to carry. This is the theory which is given by the Sutton. Now how this happens, this will be studied in the figure itself. Now in this figure mitosis, in this it is chromosome number is doubled, then it is divided. After division again chromosome number divides and multiplied, then cell divides. So each and every cell, daughter cell which is formed is having same number of chromosome. In meiosis what will happen? First chromosome numbers get doubled, accordingly then it is divided and it is divided into 2. After that there is no multiplication or doubling of chromosomes. So only 2 daughter cells have been formed, which is further divided to form 4. All 4 are having half genetic material, which further divides to form different more genetic material or in a complete form. Okay. Now in this theory of inheritance, maximum work was done by Walter Sutton and Theodore Boveri in the year 1902. He has given the theory regarding the germ cells which is being generated during the process of meiosis 1 and meiosis 2, which we can easily understood with the help of the figure which tells you how the germ cells forms genetic material and which have possibilities, various kind of possibilities in which content or chromosome is independently assorted. Independently assorted means that they follows the same law of inheritance which was given by the Mendel. Like in meiosis first you will see clearly in anaphase first, meiosis 1, anaphase 4, both are having different genetic material. When this process comes to anaphase 2, then the segregation gamete formation is there. During the gamete formation, the centriole divides and forms a germ cells. So what is the meaning of germ cell? Germ cell means a single individual genetic material strand, that is chromosomal strand, which is independent. Whenever it will mix with another, it will carry out all the characteristics or component or information through it. As we all know that we are having much more similarities between our parents by physical genotypic or phenotypic, we are having the similarities. So who is basically responsible for it? These chromosomes are responsible, because these chromosomes are the carriers, and this figure itself explains that how they are independently assorted. Independently assorted means they are not mixed together when they are together in a form of in a cell. When the chromosomes are together, still they are not mixed, they are carrying their information segregately. Whenever they get the chance, they will form another content. This difference we can easily understand by this table, that is Mendelian inheritance and meiosis. Meiosis is a chromosomal behavior and Mendelian inheritance is factor inhibited. So in the first it is given monohybrid cause F1 generation. So it is always going to be a dominant one. The dominant characteristics will show it and exhibit its characteristics. Same happens with the meiosis as well. It shows independent behavior. If it is studied under the microscope, cell division takes place meiotically, and it shows that chromosomes are very much independent. Okay. In Mendel factors, it makes stability with a number and structure and function. And chromosomal, it also do the same. In Mendelian, whatever the factor presents, that is allelic pair segments, they are formed during the times of gamete formation. And in a chromosome, they are homozygous, means similar characteristics. They form synapses first, then they divide accordingly in aphrophis first and anaphis first in the meiosis. Okay, Mendelian factors of their factors are represented with the dominant characters like capital R, small r. But zygote, meiosis first, the chromosomes can be expressed in the form of heteromorphic. Heteromorphic means both will be the different, like in a sex chromosome X and Y, they are together. Okay, in Mendelian they are segregated independently, they are segregated. But in a chromosomal behavior, their bivalent parents are there, hence the chromosome number will be bivalent. Okay. Now this is what we have studied in chromosomal behavior of inheritance. Now this chromosomal behavior of inheritance we can find in sex determination as well. So what is sex determination? Sex determination is the identification of species from the parent, how new generation or genetic material is being transferred from one generation to another through allosomes or chromosomes. So what is allosomes or chromosomes? There are two types of we can say chromosomes can be divided into two groups, autosomes and allosomes. Autosomes are 22 pair which are similar in things, pair and characteristics, but sex chromosomes, they are quite different. Basis on it, as sex chromosomes are different in males and females, we will have certain studies. This follows. The first term will be is males are maximum cases are heterogamety. What is the meaning of heterogamety? Heterogamety means the pair of chromosomes which they are possessing are different. Whether you take a case of insect, butterfly, Drosophila, insect, whatever the species you will take, frog, cockroaches, human beings, males are heterogamety. Maximum case it is found that males are heterogamety. Heterogamety simply means they are having different pair of chromosomes. That is known as heterogamy. Okay, many insects in chromosomal differences between the sexes. Many are having, but just because of their chromosomal differences, they can be heterogamety or homogamety. Okay, so males are heterogamety. Now where can be female heterogamety? In cases where females are having different pairs of chromosomes, then they are considered as a heterogamety. Okay, like some words include domestic fowl and some fishes. They are heterogamety because they are having different gene with the in this case males are homogamety because they are having ZZ, but and females they are having ZW, means one gene is different. Okay, so this is very much clear. Okay, heterogamety and homogamety. In humans, males are heterogamety and females are homogamety. Females are why females are homogamety? Because they are having XX chromosomes, similar chromosomes, but in male they are having and that is heterogamity. That means they are having X and Y chromosomes. Now coming to this, now we will be doing sex determination in butterfly. Now in this you can see males are having AA chromosomes. Okay, males are means homogamy. Males are homogamic and females are heterogamic. Okay, because males are having same pair AA plus ZZ, but if females A is there, Z O stands for there is no any chromosome, absence of chromosome. So in this case, who will decide the sex of a offspring? Male, because male are because female are heterogamity. Whatever the species, whether male or female, whosoever is heterogamity will determine the sex of the offspring. Okay, now in this figure you can easily see which can be. In male they are forming a gamete A plus Z, similar gamete, but in female, as they are heterogamity, they are forming A plus Z and A plus O. So whenever A plus O will combine with A plus Z of a male, as male is homogamic, so the product will be female, because in this case females are heterogamic. Now coming to human, in humans who is heterogamic? Male are heterogamic. When male are heterogamic, that simply means sex determination will be followed by the male itself. Okay. Now we supposed to go for the next part of chromosome, that is linkage. Linkage is considered as the inheritance of a gene. It's an inheritance of a gene of the same chromosome together, and capacity of these genes to retain their parental combination in the subsequent generation is known as linkage. Then what is linkage? Both gametes or chromosomes are together, but one of the it will be having the capacity to subsequently regenerate or generation of combination of parental in the upcoming or F2 generation. Okay, this study was done by Batson, Saunders and Punnett in 1905, and it can be well understood by this figure. This figure is very much you can come in exam. Now in this, parents are given female and male, yellow and white. Do study the structure. There will be a bit confusion. Female yellow white is having Y and W initials, but male, as they are heterogametic, be clear, it's a heterogametic, they are having Y dash and W dash and black strand. Okay, in this case female are having white miniature means W and M, but male, as it is a heterogametic, it is having W dash and M dash. Okay, now they are forming gametes. Gamete is been formed. Now this is an F1 generation. When F1 generation will cross, then you can see the wild type, first one, wild type, it is having YW and Y dash W dash, means crossing over is there. Okay, now we move downward and we find parent type is having this much, 98.7% of the old one, and this also, when subsequently it will divide and form, gamete formation will be there, and at the end you will find, when it comes to the F2 generation, the same gene retain its parental constituents, that is yellow and white, when the cross is being made between them. This might be looking a bit confusing, but if you make a checkerboard that I have told you for the dihybrid cross, this will come true. This is according to the laws of inheritance. Okay, so just go through the diagram, whatever the doubts you are having, be patient, ask queries to me. You know my WhatsApp number, you can contact me, you can call me. Okay, and make sure you will understood this. If you are having any doubt, I can repeat the lecture, I can give make a new slide for it, so that your doubts will be much more clear and you will understand the phenomena that chromosomes are also responsible for carrying the genetic information from parent to young one. Okay, this is all for the day. Thank you very much.